Morning, good morning. If I just get let you in, good morning. We are in Canvas looking at the unit genetic disorders and karyotyping. If you would read that while we wait on some other folks. Good morning, good morning. All right, so again, good morning. And we are in Canvas in the unit genetic disorders and karyotyping. If you would read that over for the next few minutes while I get all these other things set up, I'll be with you in just a few. This is also our last unit in this module, so we'll be looking to review um, Friday when I see you next. Tomorrow, we'll do some practice problems with um, actually, we'll probably just make sure we review the worksheet that you're going to do today because we're going to do karyotyping today. So we'll probably do some progress learning activities on karyotyping. So give that a good look over there. Quite a few things on that page. There's some videos to watch. And we'll watch the first one with you. Then I have a second Amoeba Sister video that I want to share with you. If you have any outstanding assignments, you want to make sure that they're in by tomorrow. Progress reports are going to be able to view on Friday. So you want to make sure all of your grades are up to date if you have anything outstanding. That's with Maxwell. All right. Please read this section before you try to look at the worksheet. Read this section over first. Okay, looks like everybody's here except Maxwell. All right, so again, we should be in Canvas under this module under genetics, disorders, and karyotypes. Genetic disorders and karyotypes. As soon as I finish attendance, I'll be right with you. Okay. Okay. All right. So everybody should be here with genetic disorders and karyotypes in the introduction lesson. Um, the video that I'm going to show with the amoeba cyst is a good recap as far as what chromosomes are. And then it goes on to talk about karyotyping in particular. And then we'll come back here and look at disorders. What happens when you have genes that are not lined up the way they're supposed to line up. And this all happens again when we did mitosis and meiosis. All of these things happen during that part of the cycle. So when you don't have the separation and matching that you're supposed to have, then, excuse me, you end up with genetic disorders. So I want to share this video first. So again, they're going to start off with a slight recap of what chromosomes are.
Many pictures of chromosomes. Well, diagrams really. Diagrams are awesome, but I've learned I need to be careful about misconceptions that can come with them. See, in diagrams, I've seen chromosomes typically drawn in an X shape. So when I was younger, I thought, okay, all chromosomes are an X shape, which was really confusing when I heard people could have two X chromosomes or an X and a Y chromosome, because then I started to wonder, why are these chromosomes resembling the letters of the alphabet? And then sometimes they look like single stick pieces and not X shapes, and I wondered what is going on with these things. I also thought that if humans have 46 chromosomes, then they're in there somewhere deep inside the body, that if you need to analyze them, you'd have to find them and make sure not to do anything to them because you only have 46 of them. Well, these are some misconceptions I had. We're hoping to clear up a little bit about chromosomes. Now, our illustrations are not scientific, obviously, but they can help with our explanations, which are. While you can find chromosomes in bacteria where the chromosomes tend to have circular shapes, we are going to focus on eukaryote chromosomes. Specifically, we're going to focus on human chromosomes. Chromosomes are made of chromatin, and chromatin consists of DNA and protein. A chromosome itself is really intense packaging. It starts with DNA being wound around proteins called histones. This forms nucleosomes, which are often considered to look like beads. More and more condensing packaging levels occur. We highly encourage you to explore this until it results into the chromosome level. But why all this packing? Well, in your cells, you have a lot of DNA and it has to fit inside a nucleus. Another reason it's also really important for when you make more cells, like in the process of mitosis. But in mitosis, you have to be able to move DNA into new daughter cells. You have so much DNA that these highly packed chromosome units make it easier to do so. By the way, it's not always packed up so tightly. For example, in DNA replication, DNA typically needs to be unwound. There are a variety of factors that also influence how tightly packed chromatin is. Let's get kind of a general idea about how human chromosomes may be represented. Human chromosomes can be like this as a single component, or they can be replicated, like this. In our chromosome counting video, we mentioned when chromosomes are duplicated during interphase, the newly made chromosome is still bound here in this region called the centromere. By the way, this region does not technically have to be in the center of the chromosome. Because of it being joined here, we still count this as one chromosome unit. But that can be kind of confusing. We say that there are two sister chromatids. So one chromosome here, it duplicates, and now, while this is still attached, it's still one chromosome. So in humans, you have 46 chromosomes. In interphase, they are duplicated. There are still 46 chromosomes here, but that's 92 chromatids in the picture. During a stage of mitosis, those 92 chromatids get pulled apart, so each cell will have 46 chromosomes. Now, just a side note, while most of your body cells have 46 chromosomes, it's important to remember that certain genes on the chromosome may be turned on or off by a variety of factors. For example, you wouldn't want your eye cells actively using a gene to produce stomach enzymes. That'd be a problem. So, would you ever get a chance to see all 46 of your chromosomes clearly? Well, you could in something called a karyotype, an image of all your chromosomes. It's possible to have a karyotype made as part of some types of medical screenings. In a karyotype, the chromosomes are stained and visible. The chromosomes from a karyotype are typically from a cell that is in a mitosis stage, typically metaphase. As chromosomes are condensed and thick, a karyotype during interphase would be a challenge because you don't have that condensed DNA packing. Now, something confusing to clarify. When searching for karyotypes, sometimes it'll be represented kind of like this diagram, and sometimes you see something like this. And while both show 46 chromosomes, the number of chromatids in the two images here seem different. So, what's going on? Now, while the mitosis stage is anaphase or telophase would be a time when each chromosome would have a single chromatid, those phases are generally not when you do a typical karyotype. For optimal viewing, a typical karyotype is taken at or right before metaphase. Recall from mitosis that in metaphase, the chromosome would have two joined sister chromatids. It's just that many times the joined sister chromatids of the chromosome can be so close together that to an untrained eye, it might be difficult to see that each chromosome in the karyotype technically has two sister chromatids. When arranging the karyotype, the chromosomes are arranged in homologous pairs. Homologous chromosomes are about the same size and contain the same types of genes, and in each homologous pair, you receive one chromosome from one parent and one chromosome from the other parent. 23 pairs. 22 of the pairs, 44 of the chromosomes, are called autosomes. That means they are not related to your biological sex. They may have genes related to eye color or height, or hair texture. The last two chromosomes are called sex chromosomes because they determine biological sex. Females have two X chromosomes, males have one X and one Y chromosome. Oh, and just a side note, the name X and the name Y have nothing to do with the shape of them. The reason is an interesting topic to look up, though. So 23 of these chromosomes are from the father, from a sperm cell, which is a gamete. Unlike body cells, gametes have half the number of chromosomes as body cells. Sperm cells are considered haploid because they only have one set of chromosomes. The other 23 of the chromosomes came from the mother, contained in an egg, which is also a gamete. An egg cell is haploid because, like the sperm cell, it contains only one set of chromosomes. When a sperm and egg cell combine, they form a fertilized egg, known as a zygote. The resulting cell is diploid, as it contains two sets of chromosomes, and it will divide to continue to form more diploid cells. You are a diploid organism. Now, if you look at the sex chromosomes, you know that mothers can only give an X chromosome, because that's the only type of sex chromosome they have. But fathers could give an X, the baby would be XX, female, or a Y, the baby is XY, male. So fathers determine the biological sex. So why do we care about karyotypes or learning about chromosomes? Well, one reason is it can really help when we are trying to understand genetic disorders. A karyotype can reveal missing chromosomes or extra chromosomes at specific locations. Do you have an interest in a career that involves this area of study? The field of genetics continues to expand, and the career of a genetic counselor may be something to read up on. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay. All right, so they said a lot of things in there that we need to unpack today. So see who was listening. We're going to look at karyotypes today, and you can't look at karyotypes during any phase of mitosis. What phase do you typically see the chromosomes in that look like this? What, somebody type in the chat, what phase of mitosis would you find the chromosomes so that you could map them out like this? See if you were listening. Bonus points on the next quiz. Okay, kind of a recap. She said that if in order for us to actually view the individual chromosomes so that they look like this, typically in the nucleus, they're all wound up like spaghetti. But during mitosis, Thank you, Malik. Malik gets in there. Natalie, you're all tag team there. Okay. They both said during metaphase. So during metaphase, the chromosomes line up in the middle. Okay, so they have shrunk because when you first start off with mitosis, you still have the spaghetti and they have to shrink and form chromatids. And then during metaphase, they line up. So in order to obtain the chromosomes so that they can make a map, they take a cell that is in the metaphase stage. All right. The other thing you need to remember that she was talking about in the beginning is misconceptions, that every cell in your body has 46 chromosomes, not just a particular cell. Every single cell in your body, for it to do what it's supposed to do, has 46 chromosomes. And the last set, and I don't know if you can see that I'm circling that on the one that I'm showing here. This is they had two on here, and it really doesn't look like this. You're either going to have an X or a Y. You're not going to have XY, and XX will not be in a normal carrier. You're going to have, notice it says one or the other. Okay. On the other side, you have one or the other. Okay, so that's one thing you need to remember. All right, so let's look at the disorders first, and then we'll look at the worksheet that I want you to work on.
An overview of some common genetic disorders. Dominant allele disorders. Achondroplasia is a dominant condition in which the long bones do not develop properly, resulting in unusually short arms and legs, a large head, and a relatively long torso. Most cases of achondroplasia occur spontaneously as the result of a new mutation in an egg or sperm of one of the normal-appearing parents. Only 1/8 of the children born with this condition inherited it from a parent. Huntington's disease is a progressive degenerative disease that can cause certain nerve cells in your brain to waste away. As a result, you may experience uncontrolled movements, emotional disturbances, and mental deterioration. Huntington's disease is inherited as a dominant allele. If one parent has this disease, the offspring have a 50% chance of inheriting it. Recessive allele disorders. Cystic fibrosis is an inherited recessive disorder resulting from a mutation of the gene that regulates the secretory gland, including mucus and sweat glands. The characteristics of cystic fibrosis are salty tasting skin, normal appetite but poor growth, and poor weight gain, excessive mucus production, frequent chest infections, and coughing, shortness of breath. Both parents must carry the allele for it. Albinism is a recessive disorder that occurs when one of the several genetic defects makes the body unable to produce or distribute melanin, a natural substance that gives color to your hair, skin, and iris of the eye. In one type of albinism, people have white or pink hair, skin, and iris color, as well as vision problems. Another type of albinism, called ocular albinism type, affects only the eyes. Sex-linked disorders. Hemophilia is a disorder that impairs the body's ability to control blood clotting, which is used to stop bleeding. Because it is X-linked, only males typically exhibit symptoms. Muscular dystrophy refers to a group of X-linked inherited diseases marked by progressive weakness and degeneration of the skeletal or voluntary muscles that control movement. Duchenne muscular dystrophy is the most common and severe childhood form of the disease. Color blindness is an X-linked condition that results in the inability to perceive differences between some of the colors that others can distinguish. There are several types that enable some individuals to perceive different colors than other affected individuals. Some cannot distinguish between red and green, while others have the inability to distinguish between blue and yellow. Chromosomal disorders. Klinefelter syndrome is a condition that occurs in men who have an extra X chromosome in most of their cells, XXY. The syndrome can affect different stages of physical, language, and social development. The most common symptom is infertility. Because they often don't make as much of the male hormone testosterone as other boys, teenagers with Klinefelter syndrome may have less facial and body hair. Turner syndrome, a rare disease that only affects females. It is caused by the abnormality in one of the girl's X chromosomes, leaving her with only one fully functional X chromosome. As a result, girls with Turner syndrome tend to be sexually undeveloped and infertile. Other common symptoms of Turner syndrome include a short stature and a webbed neck. Down syndrome. Children with Down syndrome have some of the following physical traits: short stature, weak muscles, and a short, wide neck with excess fat and skin. Small, low-set ears, an irregularly shaped mouth and tongue, and mild to moderate mental disability or retardation are also characteristics of Down syndrome. An extra 21st chromosome is responsible for this disorder.
Okay. All right, what I want you to do is go to the next page. We're going to look at the worksheet. We're going to come back to doing those problems, but I believe yours is in the module. Yep. All right, so if you go to the next page, that's where your assignment is. And if you download that, all right, so we want to download this worksheet and we want to answer it. All right, so you've got two sets here, so make sure that you know that there's a distinction between one set for questions 1, 2, and 3, and then there's another set for questions 4, 5, 6, and 7. All right, although they do look all together. Notice they number the chromosomes. When karyotyping is basically chromosome mapping, and what they do in the mapping is the reason for the numbers is that they know that certain characteristics are found on certain chromosomes. So chromosome number 1 has a certain set of genes on it. Chromosome number 10 has another set of genes on it. So they map them. They find the ones that are alike and they put them together and they number them. And they don't number the last one. The sex ones would be 23, but they just either label them to be the XX or the XY. So that's the numbering. All right, so let's look at the questions. And I'm going to do questions 1, 2, and 3 with you. And then I'm going to leave you to do the rest. Actually, I might do all the way to 7. So has everybody got the worksheet downloaded? Everybody ready to follow along? Okay. I'm not seeing any no's, so I don't want to switch to GoGuardian and make sure y'all are there. So, but if I do, I should see everybody with a worksheet pulled up. And again, if you're doing these in class, you shouldn't be behind on the assignment because this one should be finished today and submitted today. So it shouldn't be late. Because some of y'all log in and try to do other things in other classes, and then you don't have this one done. All right, so question number 1 says, look at the karyotype to the right. Circle the autosomal chromosomes. So what you want to do on your worksheet, and I can't do it on mine because this is just a copy, is to circle everything 1 to 22. You want to put a circle around all of that. 1 to 22 should be circled. The next part says put a box around the sex chromosome. So on this one, it's XY. So you want to put a box around your XY. Okay. All right, you following? Everybody following? Everybody doing? All right, the last one says, is this person male or female? Well, there's an XY, so is this person male or female? You can type it in the chat. What is this person? And then again, put it on the line for your question. If this is XY, what is this? Thank you, Natalie. This is a male. All right, so let's look at question number 2. How many total chromosomes does this person have? Now, it does say total. When you read these questions, you have to be careful. It says total chromosomes. It doesn't ask you how many pairs of chromosomes. It says total chromosomes. How many would you say? Somebody can type it in the chat. How many would you say? Thank you, Malik. 46. So make sure you circle that one. Thank you, Krishan. All right, it says put a star on a homologous pair that anyone. So again, vocabulary words. What is in a homologous pair? Homologous pair means that they are coding for the same type of information. One from mom, one from dad, but they're coding for the same type. And right now you can't. Okay, we're not talking about homozygous. So this is homologous. That means that both chromosomes, this chromosome has the same information as this one. And it might have several types of, and it could be dominant or recessive. Like you said, dominant 2, dominant or recessive. But it means that you can put a star on any one of those numbers. On number 2, number 3, anyone is a homologous. They are lined up with matching genes that have matching traits on it. So that's a homologous chromosome. So make sure you understand that definition. That's different from being homozygous. Homozygous has to do with the genotype of the individual. That's not the same as a homologous chromosome. Okay, so again, you're going to have to make sure that you review your vocabulary. Okay, so there's a difference. So you can put a star on any one of those. So now you have a circle around 1 to 22. You have a box around the XY, and you have a star on any one of those pairs. Okay? All right, it says look at the chromosomes. So now we're on the next set. It says look at the karyotype to the right. Do you see any chromosomal abnormalities? Look at it closely, and what do you see? Somebody type in the chat what you see that is abnormal. I'll give you a hint. There should be chromosome pairs. One of them is not. One has 3. Which one? Thank you, Natalie. If we were counting the X. Okay, the X chromosome has 3. It should only have 2. It should be XX. So you want to say yes on that one. There's an abnormality. Put a circle around the abnormality. Number 5 says circle the abnormality. And it says using the key below for chromosomal diseases, identify the disease this person has. So there's a chart here. This person has 3 X chromosomes. So what do they have? What's the name of the disease that they have? Triple X disease. Thank you, Natalie. All right, so you want to make sure that you put that on the line for number 6. Is this person male or female? Well, they only have X chromosomes. They don't have a Y chromosome. So this person is female. Thank you again. Number 6 is we came down to the chart here and we looked for the 3 X's and it was triple X disease. Triple X disease. And because it's only X chromosomes and no Y chromosomes, number 7, the answer is female. All right, where did Landon go? Just left us. All right. So I'm going to let you continue working on this worksheet, but that's generally how you should approach this, okay? So you have 8 through 12, you have a chart, and then you have different structures down here, but you're also supposed to use your chart up here to answer that, decide whether they are male or female, and identify the disorder. Okay, so use the rest of the time to complete this worksheet. If you finish that, complete it, upload it. Oops, I didn't mean to do that. Okay, let me come back this way. Come back, come back. If you finish that, go back to the module and finish the activities with karyotyping. If you have any questions, please don't hesitate to ask. Okay, so if you finish with that, make sure that you do the challenge before you go. I'm going to do a quiz, and typically I take the quiz from the challenge before you go. I'm also going to add the vocabulary words that we talked about earlier, such as homologous, autosomes, haploid, diploid, those kinds of things. So if you finish early, please use the time to review vocabulary. All right, again, the objective is to get this worksheet completed and turned in today so that it's not something that's going to hold you back. Again, progress reports are due this week, so we don't want to have any missing assignments showing up. And also, this is our last module or last lesson in the module. So we're coming up on a test. So make sure you get all of your worksheets completed. All right. Time is almost up. If you have any questions, anybody have any difficulties, please make sure those worksheets are uploaded. Like again, tomorrow we'll have some quiz questions to work on um, from Progress Learning again. And so make sure that you're up to date on everything else. And again, progress reports are going out on Friday. So if you have anything that's outstanding, please make sure you catch it up. That being said, if you have no questions of me, you're free to go. Wow.